Hi everyone. Yesterday I have gone through a shocking news that after 42 days, a patient from Kerala is still COVID positive. Why it is happening so? I am going to discuss about that factor. That is because of viral nucleic acid shedding, the particular process happening. Whether the viral nucleic acid shedding is a problematic thing in COVID-19 infection. This is what we are going to discuss. First, we need to know how our immune system is going to act upon the virus. Usually, a lot of my friends ask me the question, there is no medicine for COVID-19. How it is going to, it will be still in our body or it is going to be removed by some mechanisms of our body. Many friends of non-biology background ask me like that. The thing is that our immune system, we have an immune system consists of large number of cells. As this COVID-19 enter our body, some of our immune cells, a lot of cells are there. I think you people are known about white blood cells, the cells which belongs to this white blood cell category. Some of these cells will produce antibodies. Antibodies are proteins. These antibodies will bind to these viruses and aggregate it or agglutinate it. It will aggregate the virus in a particular area in the blood. As we see in this picture, as we see ants used to pull this sugar particle towards their home. Like that, consider this sugar particle as a virus. The ants are the antibody. The antibody will prevent these viral particle to move to the lungs or intestine, which result in causing infection. So it will prevent the movement of this virus in the blood. So it will block or it will prevent the movement by aggregating all virus in a particular area, so that some other cells of our immune system will come and destroy this virus by cleaving its envelope protein and killing the virus. So that is the process occurring, one of the process occurring while killing the COVID-19 virus. So our body has its own mechanism to kill the virus. It will take 7 to 12 days for this preparation of this antibody and the killing whole mechanism. It will take about 7 to 12 days. In more severe patient, it will take up to 14 days. So only we are saying to take incubation up to 14 days. After 14 days, the virus will be purely killed from your body and you will be free from the virus. Another mechanism also we have, our body contain another cell called T cell. This cell usually, if our lung cells get infected, 2-3 lung cells get infected by the virus, this T cell will come and kill the cells which are infected only and thereby protecting the other cells. These are the two types of mechanism how our body act upon these COVID-19 viruses. So, by antibody as well as by T cell, our body can kill the viruses. So, don't need to worry. By 7 to 12 days, our body will generate. If your immune system is perfect, I am saying one more time, if your immune system is perfect, the body will kill the virus either by antibody mediated or by T cell mediated mechanisms. So as the body will kill the viruses, the virus will virus as the nucleic acid, virus as the genome, it is an RNA genome. Its RNA is not degraded by our immune cells. So this RNA will be released into your body fluids, blood, etc. Usually during in our COVID-19 test, usually we take the body fluids and we will do a real-time PCR. For real-time PCR, we will isolate the RNA or the nucleic acid or the genetic material of the virus and check for whether it is the virus, whether the RNA belongs to the virus or not. That is what we are checking now. The COVID-19 checking is going on in India. So our body has killed every viruses by these 12 to 14 days. I have said, as our body killed the viruses, our RNA is get released. This RNA will remain in our body for another from the infection to 6 to 8 weeks in our body. It's from the research articles I am saying 6 to 8 weeks. Some are saying 4 to 5 weeks, like that. Anyway, 6 to 8 weeks the RNA still remain in our blood. So 6 to 8 weeks, it's around 60 days. Around 60 days the patient may be checked for as corona positive. Because its RNA is still present, we will isolate the body fluid and do the same RT-PCR or real-time PCR, then we will get the corona positive result. But the patient is, after the 14 days, is free from the virus. Virus is killed by his or her body. Now only the RNA is present in her or his blood. So this RNA is not capable of any infection. So that patient is free from virus now. The patient is still corona positive, but the patient is still free from the virus now. He or she can't infect any other people. So these type of people, after the 14 days incubation, if it is positive, it is due to the process called viral nucleic acid shedding. Not because of the presence of virus. We don't know whether the secondary infection can occur or not. For confirming this, we need to do viral culturing. If we do viral culturing, we will get to know whether the virus is inside or the RNA which is shedded by the virus when it is get killed is present in the body of the individual. I had just made a picturization of this phenomena. We can see here the SARS COVID-19 virus is in the blood and it is getting neutralized by the antibodies bound to this. This is the antibodies Y-shaped molecule. This will bind to the SARS COVID-19 and get neutralize it or aggregate or agglutinate it, so that it will aggregate the viruses in the blood, so that then the immune cells, the cytotoxic cells, will come here and engulf the virus and dissolve its membrane and kill the virus. So now the person is free from the virus. The patient is negative to the COVID-19 virus. But these process take place about in 7 to 12 days. Usually in critical patients it will take around 14 days for all this process to occur. After 14 days also, as the virus here is got killed, it will release its nucleic acid, its RNA. This process is called as virus shedding. Usually we use the PCR, that is the real-time PCR, for detecting the presence of COVID-19 in the blood. So this RNA is still present in the blood, but the virus is completely destroyed from the person. The person is non-infectious then. But the RNA still remain in the body fluids. So if we check using real-time PCR, the patient will be positive to COVID-19, but he or she will not be infectious. Only the RNA is present in the person, so the person will not be infectious. This process is known as the viral shedding. So there is nothing problem regarding the viral shedding in concern with the infectivity. Usually the person with viral RNA without the virus is non-infectious in nature. So it can be detected using this real-time PCR. The patient is non-infectious. Usually viral culture is required to confirm this viral shedding, whether it is viral shedding or not. This is the same thing which happened during the cytotoxic T cell attack the cells infected by the virus and kill the cells. Then also the same RNA will be released, and these RNA will still remain in the bloodstream or body fluids, which is detected by the RT-PCR, but the patient still will be without the virus. The whole virus will be, patients is still be free of the virus, but the patient will show the viral positive. This is just because of the PCR will detect the shedded nucleic acid present in the body fluids. So viral shedding, hope you have seen the illustration. Now this is all about the viral shedding. This is all about why the patient after the infectious cycle of 14 days still positive. Because of this viral shedding, the patient remains positive. I hope you people understood the process of viral nucleic acid shedding. Hope you enjoyed this video. Please share this information to more people so that in order to prevent fake news spreading throughout the country. So this is all this data which I am saying here is taken from authentic research papers only. Thank you for viewing. Have a nice day. Thank you.